Okay, so my name is Dominic Papineau. I'm a postdoctoral research associate at the Carnegie Institution of Washington and affiliated with the NASA Astrobiology Institute since 11 years now. And my research addresses questions related to Precambrian biogeochemistry as well as the evolution of early life, specifically targeting microscopic mineral assemblages as possible biological signatures that could eventually serve as a model for looking at samples that will be returned from Mars and maybe looking for these specific mineral associations with the kinds of microanalytical techniques that are now available in the field of early Earth and Precambrian geoscience and so on. The role of phosphorus in life is very well known. Phosphorus is an element that is present in nucleic acids like DNA and RNA. It's part of proteins, where proteins get activated or enzymes get activated, deactivated with phosphate groups. It's part of lipids and lipid bilayers, where phosphates are an intrinsic part of these phospholipids that compose the bilayers of cells. So it is really an important element for all life forms as far as we know, and therefore it must be preserved somehow in the geologic record, and the way to look for them is to look specifically at phosphorites and at minerals like apatites that can form from the organic matter that ultimately in the sediments get regenerated, oxidized, the phosphate is liberated and can be converted into minerals. Over geologic timescale, phosphorus accumulated in coastal environment wherever the conditions were such that it could accumulate, but it stimulated blooms of primary productivity, blooms of oxygenic photosynthesis, in a way that's analogous to what we're seeing today. I mean, it's a very important rate-limiting nutrient, and over geologic timescale, it is the most important rate-limiting nutrient. And in terms of blooms of oxygenic photosynthesis, we see this in eutrophic estuaries today. And in the Chesapeake Bay area, in the spring, the agricultural runoffs that deliver all this nitrate and phosphates, more importantly probably, ends up stimulating these algal blooms. It makes for anoxic conditions in the water column, creates a lot of biomass, and in probably a similar way, this is what happened when the phosphorite depositions of the Paleoproterozoic, but also that of the Neoproterozoic Lower Cambrian occurred. We're actually exploiting the phosphate accumulation of the rock record that was basically happening under conditions of cyanobacterial blooms to stimulate today again growth of agricultural products like corn and various other things that we grow in our crops. So we're using it twice. The phosphate is being reused and reused over and over again over geologic timescales, but today it's anthropologically driven.